Hey everyone, thanks for checking out my talk here, and also to the organizers of this workshop for inviting me to share my thoughts on sampling organic matter in tropical environments. So I think my presentation will probably overlap with a few others in this workshop, but I'll give you a general overview just for completeness. What I'll be doing, I'll share some brief considerations for working in tropical environments, and then I'm going to focus on a case study of mangrove environment, which is my specific research expertise. Understanding coastal change using radiocarbon is typically done in a depositional environment. So in the tropics, some of these environments might be lagoons, mangroves, or beachrid systems. And to really understand what type of things you should be sampling, you need to relate that to your overall research question. So if I wanted to find out about past salinity changes in a lagoon, I would want to be getting an age for the past depositional surface in the lagoon. And if I wanted to find, say, the position of past sea level, I would want to get an age for the past depositional surface in the mangrove. So it's really important to think about, what event or environmental phenomenon are you trying to get an age for? So some typical things that are sampled for radiocarbon analyses are bulk organic sediment, and this is an undifferentiated, unidentified organic carbon sample. And any radiocarbon measurement on this sample type will be an average age of an unknown number of carbon sources, and generally an unknown type of carbon sources. The other key type of sample is some identifiable fossil, like a plant macrofossil or, for example, a microfossil. This could be leaves, seeds, roots, pollen, and often these might be able to be identified to a species level. And this is a measurement of one carbon type, one carbon source. So it's an age of the death of that particular part of an organism. With fossils, we need to think about whether it was produced in situ or whether it was transported, and whether they're long-lived or short-lived. So, for example, will the whole fossil have the same age or not? For example, timber has different parts of the wood that might have different ages versus, say, a leaf that only lives for 6 months of the year. And at the end of the day, how do each of these sample types relate to the environmental phenomenon of interest? Some general considerations for working in tropical environments are that, these environments are characterized by high rainfall, temperature, and humidity. And this type of environment means that you actually have really high production of organic matter. There's a lot to go around. At the same time, this high production, we also have really high rates of degradation and different types of degradation pathways. But thinking again about this type of environment, are the timescales of organic matter transport a little bit shorter because stuff is more readily consumed or degraded at the surface? And all of these things can make collecting samples and interpreting samples in the field quite challenging. This is me in a mangrove, really sweaty and grumpy because a mosquito bit my eye. Good times. So I'm going to focus now on radiocarbon dating in mangrove environments, which I use to reconstruct past sea level over the Holocene. So just very briefly into mangroves as indicators of past relative sea level change. Mangrove forests typically occupy a very narrow part of the intertidal zone. In this schematic, they are between mean tide level and the highest astronomical tides. So if we collected a core in these environments or in an adjacent environment and we find mangrove sediment at depth, then we know that sea level must have been between mean tide level and highest tides at that point in time. But what point in time was that? We want to get an age on that past surface, and that is our challenge. So mangroves produce a lot of organic carbon. Per area, they're some of the highest carbon producers in the world, so there's no shortage of carbon for sampling. And a lot of this carbon is produced in situ via roots, timber, leaves, those sorts of things. And mangroves photosynthesize via the atmospheric reservoir. So you would use the atmospheric calibration curve to get a calendar age for any mangrove sample that you got a radiocarbon measurement on. So they're not aquatic plants, and you would expect carbon isotope ratios similar to other terrestrial plants. So if there's so much carbon, organic carbon, what do you sample? So mangrove plant macrofossils that come from above ground, such as leaves, seeds, those sorts of things, and that are deposited onto the depositional surface shortly after they died, would be the most ideal because they are more likely to be representative of the age of that surface. But one thing to note about plant macrofossils in mangroves and in the tropics in general is that they're usually quite big. So these are some examples from some of the mangrove sediment I've been studying. This is some bark, some seeds, and a leaf. And if you're sampling, say, with a corer, some of these sample types actually might make up a large part of the core's diameter. So it's actually, luck of the draw whether you actually sample one of these with your corer or not. This leaf at the bottom here is not actually a mangrove leaf. They're typically much bigger than that, greater than the size of your hand. But it's one we found in a mangrove sediment, and I'll tell you why that's special in a sec. So as I mentioned, things decay at the surface very rapidly in the tropics. So if you find a plant macrofossil in your mangrove sediment sequence, it was likely deposited soon after it died, and it wasn't really transported very far. But one caveat to this is that there are crabs, and a lot of them, in the mangroves, and crabs are really, really, really hungry. So I'll show you a little video here. So they actually consume a large proportion of surface biomass before any of it can be buried and preserved in the sediment sequence for us to radiocarbon date. And if you've ever been to a mangrove, you'll notice that the surface is actually quite clear of leaf litter. So finding any sort of plant macrofossil in a mangrove sediment is actually quite rare. There's some other fossils that have been used are pollen, so technically a microfossil that's just made up of organic carbon. And this is a radiocarbon sampling technique that has been used in a lot of other depositional environments like a marine setting, lakes and salt marshes. But these have had quite mixed success and most often result in much older ages than would be expected. It's also a very fiddly lab procedure that can easily introduce contamination, and there are a lot of papers out there that actually state that they have introduced contamination during separation of pollen concentrates. And more specific to mangroves, mangroves typically disperse pollen quite locally, so you would think that that would be quite a good thing for a study like this. They're not going to be transported a large distance. But what we found when we were studying the mangroves of the Seychelles for some sea level reconstructions is that these mangroves don't produce a lot of pollen, and even if they do, its preservation in the sediment is really poor. And this is just an image of some separated fractions using heavy liquid separation of some of that Seychelles mangrove sediment, and you can see it's just made up of a whole bunch of plant fragments and homified bacterially degraded organic matter, and there's just not enough pollen to date with radiocarbon, so that was a shame. So a big topic of discussion when using mangrove sediments for paleo reconstructions is mangrove roots. And mangroves are famous for their root systems. They have large aerial root systems that splay out horizontally, but they still penetrate a few meters depending on the age size of the tree. And obviously, the penetration of roots into the underlying sediment brings younger carbon below the surface, and it also physically bioturbates the sediment. So roots are therefore not representative of the depositional surface. So as somebody interested in reconstructing sea level, and therefore those past depositional surfaces, sampling roots are not what I want. I don't want roots. And roots can make up actually quite a large percentage of below ground biomass in mangroves. So therefore the sediment is made up of a lot of roots. This varies between sites and different sediment types and different tree types, but it's also not really that well known in the mangrove ecology community or in the carbon cycling community. And large roots, for instance, are quite easily recognizable. You can pick them out of your sediment sequence, but there are also fine roots that are not so easily separated, and these can degrade, and you might be sampling those even then when you don't want to. So if we can't get macroves and we can't get pollen and, roots are a problem, what's left? Well, bulk sediment. And most studies have used this approach for their radiocarbon dating because there wasn't much other options. So bulk sediment is made up of lots of different things in a mangrove. It's unknown what the primary source of it really is. It's probably roots, but there's a lot of work to be done on that. To the right here is a schematic of what's going on in a mangrove. Roots contributing to below ground, pollen, timber, leaves contributing from above ground. There's crabs bioturbating the sediment, and there's also the import and export of carbon and organic carbon via the tides above the sediment surface and below the sediment surface. So tying a bulk sediment radiocarbon sample to an environmental phenomenon is actually quite tricky. So what are the implications of all of this? So bulk sediments and creating our age models from bulk sediment ages typically result in poorer age models, and this is evident in the literature. But bulk sediment samples are used anyway because plant macrofossils or microfossils are not preserved. And we have bioturbation by crabs and by roots that mix the sediment and result in more age model problems. And another point I wanted to raise is the point of sedimentation rate and the generation of accommodation space. So if you don't have a lot of accommodation space being generated, if you have low sedimentation rates, then your mangrove sediment is going to have more time at the mixing interface. So there's going to be more time for root production, more time for crab bioturbation to occur, and that will change your age model results. So to illustrate that point further, I'm showing some results from a radiocarbon study we did in the Seychelles, and we collected different types of samples from the same elevations across a mangrove in the Seychelles. So the different types of samples we used, we had bulk sediment, macrofossils, and organic concentrates, which are a sieved sediment fraction, which I'll go into a little bit more in a minute. And these are shown by the different colors we have here. And each bar that you can see is a calibrated age range of a sample, the 95% confidence interval. And you can see that the ages vary quite a lot. And this is because the Seychelles has had a relatively stable sea level over the past 3,000 years or so. The mangrove sediment accreted very rapidly initially, and then just sat around getting continually mixed for quite a long time, as there hasn't been any new accommodation space generated, and that's why we have such a variety of ages. In contrast, this is an age model from a mangrove core we collected in Micronesia, where we have nearly 30 radiocarbon dates on sieved bulk sediment, and this gives us a seemingly sensible age depth model. At this site, there is localized subsidence, which generates continuous accommodation space for the mangrove sediment to keep accreting, and therefore the mangrove sediment sequence doesn't remain in the mixing zone for an extended period of time compared to the Seychelles. So some working solutions for this: sample surface macrofossils, if you can find them. This is a study from a Belize mangrove that constructed their radiocarbon ages from leaf fragments. So it is possible, and it shows here that it actually results in a pretty consistent age depth relationship. And this will require, careful core examination and identification. Another working solution is to use a specific sediment size fraction for radiocarbon measurements in hope to remove some of those larger roots, the root contamination, and to hopefully isolate something that's more reflective of the depositional surface, the age of the depositional surface. So this is a study from a mangrove in Zanzibar in Tanzania that compared bulk sediment ages here in black, the little black squiggles, versus a sieved sediment fraction, which is in red. And the sieved sediment fractions are typically older than the bulk sediment ages, and they tend to have a better age depth relationship than the bulk sediment ages. But this pattern varies across sites. So we tested sieve sediment fractions versus bulk sediment fractions for radiocarbon ages in the Seychelles mangroves, and we found that there's little to no difference between these two sample types. These plots here compare older versus younger age probability distributions, and the only difference that is greater than the sample measurement error is the macrofossil samples. So in the Seychelles, the bulk sediment ages and the sieve sediment ages are pretty much the same. So some more working solutions to this, why don't you sample a larger piece of core for getting some mangrove pollen for dating? Are there other organic matter fractions that we can use? For example, compound specific radiocarbon dating. Can we use ramp pyrolysis techniques to understand which carbon fraction within the mangrove sediment is best to use? I think there are some other talks on these two topics in this workshop, so you should go check those out. And another thing that I'll just very briefly talk about, because it's quite contentious, is the pre-treatment of organic carbon prior to conversion to graphite. Do you do acid-base acid or not? For some mangrove samples, doing a traditional acid-base acid seems to remove a lot of carbon that makes age models make more sense. So the question is whether you just acid treat them or base treat them or not. So mangrove samples generally require a bit of trial and error to know which samples to use, and it will be different between different sites, and I recommend doing a little bit of that testing if you want a really reliable age model from a mangrove. And obviously more ages means likely better age models. So considering your project budgets and some other workarounds to get more bang for your buck, complete some of the pre-treatments before sending them to the AMS lab, or take advantage of new techniques. For example, there's a new technique called the single-step radiocarbon process at NOSAMS. You can probably ask Roberta a little bit more about that, but essentially it means the price of a radiocarbon age is reduced, and you should be able to get more radiocarbon ages with a slightly higher uncertainty than if you used the regular approach. And at the end of the day, getting independent age constraints from lead-210, from cesium, from tephra, from pollen and pollution markers is always your best bet to improve any sort of chronology that you get from a mangrove sediment. And so a really brief summary then, consider your research question. What do you want to get an age of? Do you want the depositional surface? And what is the resolution you really want an age model at? And, so what sample is the best one to get a best result for that? My opinion is that's probably surface macrofossils if you can find them. So consider your study site as well. Is accommodation-based generation likely? Do you think surface macrofossil macrofossils are available. And are they transported or are they in situ? You probably will have to use some sort of bulk sediment sample, and if you can, get a lot of radiocarbon measurements. And again, trial and error, every site is different. Do some testing. And I don't normally do a bibliography for a talk, but I thought it'd be useful in a workshop, so here's some of the publications I was referring to in the seminar. And please get in touch if you want a copy of any of these or if you have any questions about this. So thanks for having me.